Hi everyone, I'm Daisuke Kihara from Purdue University. Purdue University locates close to Chicago. It's about 2 hours drive from Chicago O'Hare Airport down to south. So it was my great pleasure to be able to speak here and interact with you. I have been working on protein bioinformatics, specifically working mostly on protein 3D structures. So I hope I can show you a fun part of the machine learning application in protein structure bioinformatics. So yeah, just very quick overview. There are lots of different types of bioinformatics, and the speakers of this summer school includes those people who work on DNA. But here, what I work on is the product of genes. So from DNA, it was copied to messenger RNA, and then finally we have proteins. Now, protein is the working molecule in a cell, and it has a 3D shape, it has sequence, and it assembled together into complexes. And those are those informations can be converted or predicted from one from the other. So that's what I'm working on. Yeah, and protein has a lot of have lots of information. As probably everyone knows, protein consists of 20 different amino acids. Each amino acids um, each amino acids have different properties. Okay, so amino acids commonly share C alpha, carbon, oxygen, and then the NH2, which makes the peptide bonds, and each of them have different side chains with very small ones or very long ones. Some of them are positively charged or negatively charged or have aromatic rings, and that makes the difference in the chemical properties of amino acids. So one of the things people are very interested is, is protein 3D structure prediction. In protein 3D structure prediction, you have amino acid sequence information, 20 the string of 20 amino acid letters, and then from here we want to predict how the 3D structures. 3D structure means that XYZ coordinate of each atom of a protein. We want to predict those XYZ coordinates. And here we are showing several different representation of proteins. In the first one, I'm emphasizing the volume of each atom. And the color shows the different atoms. Red is oxygen, the blue is nitrogen, gray is carbon, and white is hydrogen. So here in this representation, the volume is also like a surface shape is emphasized. The other three is emphasizing more about the chain orientation. Especially like the bottom two is emphasizing which part belongs to alpha helix. In here, in these two figures, the alpha helices are shown in green. Beta strand, where the main chain goes almost 180 degrees stretch, is showing red. To clarify the characters of the structural, the peculiar characters of this particular protein structures. So in the protein, for example, in the protein 3D structure prediction, we have input is this protein sequence, and then output is what we want to predict is the XYZ coordinate of each atom, or often also to predict which region corresponds to which secondary structures. Alpha helix and beta strand are called secondary structure of proteins. Okay, so here I'm showing the actually poster from PDB, Protein Data Bank. This protein data bank is a database of protein 3D structures. And it releases this poster. It has multiple pages, and I'm showing one of them. And here you see that this is emphasizing the volume or shape of the proteins. So there are small ones, like we see in yellow here. Those are looks like enzymes. And now, okay, you see that this big one, this is a rhinovirus. This is a virus capsid. So this is a symmetrical assembly of many pieces of the unit proteins. And it's a huge assembly. And now if you pay attention to colors, uh, you see some proteins have different colors, like this, like let's say this one, this have red piece and also blue pieces. And these are the corresponds to color is representing individual proteins. So in this case, this is assembly or complex of multiple pieces of structures. So and like okay, like this one is in the bottom. This is now the assembly between. This is a ribosome. This is assembly between of proteins and RNA. Many RNA molecules are assembled together. This is okay, GROW-ES. This is this has a cage-like structure, and it takes the unfolded, misfolded proteins in the cage and entangle, unfold it and then release it to outside so that protein can have another chance to get folded in the right way. And so and this is also a large complex with many pieces together. So from here, you can see that there's another type of question, which is protein-protein docking. In protein-protein docking, we want to predict, we have input is two proteins, either protein 3D structures or can be recently just two protein sequences. And then we want to predict the 3D structure of a complex. Yeah, so there are lots of proteins actually nowadays in the protein data bank PDB, and there are many things we can do. So here I'm showing first I'm showing how quickly the protein 3D structure data information is accumulated in the PDB database. Here y-axis is the year. Okay, starting from this graph starts from 1976, and then this is up to 2020, and this x-axis showing is number of entries in the PDB, number of structure entry in the PDB database. I think now it exceeds over 200,000 entries, and each entry may have multiple chains structures in it. So actually, if you just count the number of structures individually, you have really large number of millions of structures available. So this is a source of the protein structure bioinformatics. But nowadays, now we also have several different types of very interesting data, which the one is cryo-electron microscope. The electron microscope will give you recently a high resolution to very low resolution the image or 3D image of protein or protein complexes. So the challenge is very different because in protein structure, usually stored in PDB, has a specific XYZ coordinate information, which you can use as like a feature. But in Cryo-EM, you don't see the actual atom points. So it's more like a surface representation or a point cloud representation. And now from here, okay, so even the comparing those shapes are challenging because you don't know which points to match to which point in two shapes. And also here we also want to predict the structure, model the structure from this the 3D image constraints. That's another interesting challenge. And recently now, okay, this usually cryo-EM is for single protein complexes, but it can, the electron tomography. This is a same type, okay, it uses same type of instrument, the electron microscope, but it shoots electron to the sample, but it gives you the whole, it can give you the whole picture of 3D picture of an entire cell. Like for example, this one, this is showing, this is a bit old data from taken from a paper, but what this is showing is that this is a whole cell of an organism, bacteria, and then you can see, okay, those dots are showing the distribution, spatial distribution of this particular protein complexes in a cell. So you can see whole cell level image with very low resolution. And now, okay, so in protein 3D structure prediction field, the accuracy of prediction is getting very high, and the number of structures from prediction can be also utilized for further application, for example, drug screening, or it could be a function prediction. That's an increasing number of reliable structures being available. Now, okay, protein is not a rigid molecule. It is a physical object which is moving, which in water in a solution, and which often corresponds to the function of the protein. So people can do molecular dynamic simulation. In the molecular dynamic simulation, you can well, you compute the physical forces like electrostatic interactions or van der Waals interaction, hydrophobic interaction between every pair of atoms, and then simulate how it moves. So interaction can also include the protein against water molecule or a drug molecule, and that is a movie of how the protein structure moves. Here, okay, so here in this graph, what's shown here is how much each amino acid moves in this particular protein. So x-axis is the position of amino acids, y-axis is showing how much each amino acid moved in the simulation. This bar is showing which part corresponds to alpha helix. So in this, okay, I forgot what this protein is, but in this case, you see that those peaks here, 1, 2, 3, this corresponds to, it looks like the loop region, which does not correspond to alpha helix. So and so you have a huge amount. Well, you can okay, actually there are lots of simulation data available, and you can also do it by yourself. So that's a huge amount of data of dynamics. And of course, we are also for application, and we are interested in drug molecule. Drug molecule is also 3D compounds, 3D information. So in protein structural bioinformatics, we connect or predict those information together, and then do well, hopefully produce some meaningful prediction. Oh, by the way, if you have any question, please feel free to ask. So topics in protein bioinformatics include 3D structure. Okay, so 3D structure database search. Well, in a database search of 3D molecules, not necessarily trivial. You need to think about how to represent it, how much speed you need. So there are it's fundamental. In informatics, in the database search is fundamental, but the way to search 3D structures is not necessarily well established actually. And then 3D structure prediction is one of the hot topic even recently. So in 3D structure prediction, input is a protein sequence, and then you predict the 3D structure XYZ coordinate of proteins. And then associated problem is to predict secondary structures or disordered region. Some protein do not have a solid region, a solid structure, and is designed to be very floppy and can move around in a cell. And you can predict because there was some bias in the amino acid in the disordered region. You can make prediction based using machine learning methods. Now, okay, so this is related to 3D structure prediction, and here the structure model. Well, often the experimental biologists can collect the data, structural data, which is not for determining protein 3D structures. NMR, Cryo-EM, SACS, each of them, NMR will give you the distance between atoms. Cryo-EM will give you the overall shape. SACS will also give you the overall very low resolution shape, overall shape. So from here, how to model 3D structures is a challenging problem. Also, okay, so from structure and sequence, the ultimate goal of biology in molecular biology is to know function of the protein, and like a drug screening from structure is one of the biggest application of using protein 3D structures. And now, okay, so so I mentioned about protein complexes, but in a cell, not only the very the permanent complex, but also transient complexes uh, occurs in a cell. In a signaling pathway, protein and protein interact shortly and then pass information in downstream. And there's a techniques to determine interaction of many proteins in a cell in a large scale screening. So you have a graph here, and then from that protein-protein interaction network, you can do function prediction or drug target identification and many things. So to summarize what we have in protein bioinformatics, we have, of course, amino acid sequence and graphs. So graphs include protein-protein interaction graphs, or actually, as I will show you later, protein 3D structures can be also represented as a graph. And specifically, okay, so if you talk about evolution, you can organize proteins along the evolution in a phylogenetic tree. And we have 3D shapes and XYZ coordinates, and then 2D and 3D low images, and so on. And we have vocabulary of gene ontology, function annotation, and so on. So in protein bioinformatics, what you do is to look at those data, and then try to connect one from the other, or try to predict the one from the other. So, all right, I'll show you, as just a part of introduction, I'll show you one work we actively do. This is a 3D surfer. In this 3D surfer, this is addressing very quick real-time protein 3D database search. Typically in protein structure comparison search, you superimpose, to compare two protein structures, or any 3D objects. In principle, you have two 3D molecules, 3D shape, and then you need to because the 3D have a freedom of rotation, you need to identify the best rotation. So you superimpose it and find the best rotation, and then after the rotation is optimized, then you compute the distance of individual corresponding points. That's how usually compare two 3D structures. But that takes very long time, and against PDB, which have millions of the structures available, you cannot do the real-time search. But here, in a database search, just like a sequence-based BLAST does, or even like Google or web, or Bing, or any the keyword search does, we want to get the results as soon as possible, very quickly. So to be able to do the excuse me, very quick structured database search, we used in this 3D surfer we used the mathematical representation of protein surface. So what we did is we considered our protein structure as a molecular surface. And then I consider this as a 3D function in a 3D function in 3D space. So, and then just like similar to similar to what Fourier series does. Once I have a 3D function, I can do the series expansion, mathematical series expansion with a fixed base function. And then with that base function, I can, using the same base function for all of the proteins, I can now compare two shapes very efficiently by just computing the coefficient values in the series expansion. And for the base function, we used 3D Zelensky descriptor invariance. 3D Zelensky is also used much in the like image 2D microscope image processing, but there's a 3D version of it. It has the radial function and also spherical harmonics, and you can do the series expansion using Zelensky descriptors with different those indices. Once it is expanded, we can compute it in a rotation invariant way, which means that the rotating structures does not change mathematically, does not change as a series expansion. And then with that gives you the feature vector of a protein 3D function, protein structure, and we can compare two structures very efficiently. We will also in the next the seminar by my student Charles, he will probably address more about the 3D Zelensky descriptors because we use this also for representing molecular surface for protein docking. Okay. Okay. So let me answer one question from Filippo. So, okay, the question I see in the chat box is from PDB. Curated proteins are also putative proteins. So, right. So in PDB, most of the structures in the PDB database is kind of a very important, well-studied proteins or very important proteins. And the functions are usually known before the structure is solved because determining protein 3D structures experimentally is very costly. Sometimes it takes years to solve just one protein structures. So people do the structure determination usually for very important proteins, but there are also like until a few years ago, there was a project called Structural Genomics Project, where it's like a genome project. So it's a big project to determine protein structures without knowing functions. So in that case, so if you search PDB with unknown function, for example, or uncharacterized proteins, you will find hundreds of proteins like that, which means that region of the protein seems to be is a protein, but no one knows what the protein is doing. Also, in PDB in early days, predicted structure or model structure were also included, not recently. Okay, here I'm not sure. Okay. All right. So AlphaFold. Sure. Okay. Yeah. Of course. Okay. Yeah. All right. Sure. Okay. Maybe I can quickly answer that. Yes. AlphaFold is Google's protein structure modeling methods, which performed very well in a protein structure competition called CASP. And AlphaFold can do very good the structure prediction very high level. It's much better than the existing methods, unfortunately, including our methods. It is okay. AlphaFold is okay. So the question is, I think, asking if it is doing well in docking and simulation work without any validation modification. Yeah. So AlphaFold is designed, or at least designed, to predict single protein structures accurately. So it is not trained to do the protein docking. Um, so, um, but seems like sometimes it works for docking, but it's accidental. If you wanted to use the protein 3D structures for, AlphaFold for docking, you need to be careful. And okay, so if you wanted to use the predicted model by AlphaFold individually and then want to do docking or simulation work, um, you should pay attention to the accuracy, predicted accuracy. AlphaFold also gives you the predicted accuracy, so you need to pay attention to that. Okay. So getting more questions. How does knowing about docking help in function prediction? All right. So if you have, so the method is not that well established, but in general, if you know, I can tell you two things. One is that if you know two proteins are interacting, not the structure, but if you know that two proteins actually interact in some way, then you can guess that two proteins is related is involved in the probably same protein function. So from interaction pattern, you can guess the function of the protein by interaction pattern. Now, about docking will provide you a 3D structure of interaction. If you know the 3D structure of interaction, you might be able the docking may find you like a drug binding site, or some shapes which is characteristic for some function. So by knowing the protein docking, you have an overall shape, and from the shape, you might be able to find some features specific for certain function. But the structure to function prediction, actually, I have worked on the pocket-based binding ligand prediction. You can find some of my papers in my lab website. But that part is not that well established. It's similar to like a sequence-based, motif-based function prediction. You have a sequence, and then you want to in a motif-based function prediction, you find the sequence pattern that corresponds to a function. Here, if you have a protein docking shape, you have an overall shape, and then you may be able to identify, the function-related shapes. So there is a possibility to go from docking to function. Okay, well, let me continue. So, all right, first I'll show you the protein-protein docking. So protein docking is important because individual protein structures is being more and more available. And then also by experiment, even without knowing the protein structure, there's the large-scale experimental methods called EASE-2 hybrid system, or by combining using mass spec, you can identify lots of interacting pair of proteins in a cell in really large scale. So here, this is classical data from EAST. So in here, individual dot corresponds to a protein, and then edges indicating that these two proteins are interacting. So from here, so you know that actually from here you can collect the lots of interacting protein interaction information, and then on the other hand, from PDB, you can identify in not always of cases, but individual protein structures are being more and more available, and as one of the question was asking, AlphaFold often also structure prediction method can often provide individually reliable structures. So you can from two information together, you can do protein docking. And also protein docking is important also for artificial protein design. Here in this, this is also a bit earlier paper. In this paper, authors have assembled artificially constructed the protein complexes. And for that, so here in the design, you want to design amino acid sequence so that those proteins assemble together in a designed fashion. And for that, protein docking technique is also very important. Okay, so in protein docking, you have two structures or many more other protein structures, and then you want to do the methods usually try many different docking conformations, and then pick up the correct information, correct model. So here, okay, well, this is supposed to be the movie, but okay, sorry, I can, well, it is supposed to be a movie, but in this movie, I wanted to show the docking poses. So this is the docking between this gray protein and this magenta protein. And the correct position of this magenta is this green. And this animation was supposed to show you how this magenta protein can dock around this gray protein in the middle. And I just wanted to show you how many different, the large number of poses which can be constructed for this interaction. So later we will, Charles will show you our methods called lizard. I'll skip this, and then we will, he will do the demo of lizard web server. This is for protein docking. Okay, well, this is how to use page, but I leave it to the next seminar. And in protein docking, so also has a competition. It's called CAPRI. This CAPRI stands for Critical Assessment of Predicted Interactions. This is if you know, if you have heard about CASP, protein structure prediction, CAPRI is for protein docking. So what in CAPRI, how it works is the organizer will post question, a challenge of protein docking. So typically recently they post two sequences or many sequences which can assemble together in a complex, and then the participants like us are supposed to model individual structures in some way. Maybe now we can use AlphaFold, but we need to model individual structures and then dock them and then submit to the organizer. Yeah, so this is from, I think, 2019. Charles is here. I'm here. And the community is ready. Okay, this is part of the community who attended this meeting in EMBL in UK, EMBL EBI in UK. This was 2000, I think, 19 April. So there are about maybe 50 to, yeah, a little bit over 50 active developers of protein docking. In a protein docking, okay, so this is a general protocol of protein docking. You have two protein sequences or protein structures as input, and then now typically the method generates lots of models, because starting from two models, you can assemble the two structures in various different ways. Typically in our methods, it generates over 50,000 or even more models. So actually this is one part. So one important part, the critical part. So well, you need to generate at least well, the good models among these pools, among this pool of models. But now even more challenging is that how to select a model, because you have 50,000 models, but from there, you need to identify and rank the good models out of the bad ones. And then once you have selected maybe up to 5 or 10, then you can do more fine grid refinement, structural refinement to optimize the structures. So actually in protein docking, model selection is a key. Yeah, so here, this is, I'm showing some example of different protein docking poses. And there here I'm showing only 6 of them, but imagine that we have 50,000 of different structures. Okay, so now I'll show you 2 methods we have developed for model selection. The first one uses deep learning. Both are using deep learning, but first one is using convolutional neural network. we will go very detailed about this method, but general idea is that, okay, suppose these two proteins are interacting tightly. This is just for illustration. Then we want we get a feature. We look at the interface region and then take the information of the interface region in the by the 3D box, and then finally guess if this particular model is probably correct or incorrect, close to the correct structure or far from the correct structure. So this is a deep learning network for this task. Input is a box. Input is the input is the atom position in a box, and then output is a probability that the model is correct or is correct, close to actually is qualified as acceptable model in the Capri criteria. Well, we go this very in detail. So, okay, so let's see the input data closely. So this is one example of protein docking model. Actually, this is a correct answer. This is a PDB. If you're interested, PDB ID is 1BRS. So this is how two proteins interact. Now, okay, this is ribbon representation, but we can also, I can show you the whole atom representation. Now, okay, then I now color it by atom types. So here, red is oxygen, blue is nitrogen, and actually the those white region is carbon. So idea of this model selection method is to look at the interface, the distribution of oxygen, nitrogen, and carbon, and other atoms at the interface, and then see if that distribution of this interface, atom distribution, is likely to be correct from correct model or incorrect model. And to capture this interface region, as I showed this by the box, we put this box of certain size. Okay, the box of 20 by 20 by 20 angstrom, or 40 by 40 by 40. We tested two box size and compared which one performed better. And so we put this box, and then this box is segmented into a small cubes of 1 angstrom each interval. And then as an input feature, at each box, we count the number of oxygen, nitrogen, carbon, and other molecules in each box. And that is a feature vector, or feature information of this protein, of a protein complex model. Okay, so we will see the, okay, this is the illustration of a convolutional neural network. So we use convolutional neural network for this in this algorithm. Probably, some of you have heard about convolutional neural network, and this is an illustration I took from our website. So, okay, so in the protein here, okay, so in this example, there are two. Okay, you see that green boxes. The green boxes is showing the input data. So each box have 1 or 0, and then you can see that, okay, yellow box, small box is moving, and it has a yellow box has its own information. Like you see that 1 0 1 multiply x0 x1, so 0 1 0 1 0 1. This is a box. This is called convolutional filter. So what convolution filter does is it multiplies at each that value in the filter at each position of the input data, and then multiply it and sum them up, and then put it as a sum will be put into this convolved feature. So what it is doing is it is okay, it is capturing the neighbor information and then summarizing the information here. And by doing this process in multiple layers, you will be able to actually capture larger and the information of more broader area in an image. So in our case, this instead of a 2D image, we have a 3D box. And then we have the convolution filter of 3D box, and then we scan it. And well, this is from some earlier paper. In the initial, okay, so usually CNN have different multiple layers. In the first layer, as you can see, those the matrix is capturing the very simple shapes like horizontal lines or diagonal lines. But as you uh, stack the convolution filters in multiple layers, you start to be able to see more global view of the information, and finally it can capture large big images like faces, cars, and elephants in the feature space. Okay, let's see the chat box. Okay, all right. So let
No,
me
just
go.
technical. Yeah, it's just technical.
Yes, I saw that. Okay. Okay, so this is how the network works. So okay, let me go through it one by one. Now, okay, so by a big box placed at the center of the interface, we capture multiple things. One is, okay, the distribution, position of nitrogen. In another filter, in another box, we count the carbons at each, one by one cube in the input box. Oxygen is also considered, and occasionally there are some other atoms like sulfur. That is counted separately. In addition to these four features, we also try to consider physical energy. Um, so this GOP and IT score is a statistical scoring function which is used in structure prediction. So at each box position, we computed those interaction, energy by the atom interaction, and consider this as a feature. And this box size is 20 by 20 by 20 angstrom, or 40 by 40 by 40. Now, okay, so let's look at the big picture first. So then we put two convolution layers, CNN. There's one max pooling layer, and there's another CNN convolution layer, and then this max pooling layer. And then this is now, all these features are now spread. This up to here, this is a 3D information. And then that is spread into a one-dimensional vector. And then that is fed into the fully connected, FC stands for fully connected neural network. And then at the end, two layers. Then that will be passed to sigmoidal function, and then compute the probability that this model is correct or may be wrong. So, okay, from here to here, so the first convolution layer uses this as the convolution filter of 3 by 3 by 3. So, okay, so this 3 by 3 by 3 box is scanned from the corner of this 20 by 20 by 20 box, and then shifted by 1 angstrom, one grid point, one by one. So because it's 3, if we do this, then the size will become 18 by 18 by 18. Now, from 18 by 18 by 18, we have another convolution layer, so which makes this the size to 16. Because if you fit a 3, the box of 3 angstrom to 18 angstrom, you lose one size. It's becoming smaller size. Now from here, now this is the next layer is max pooling layer. Max pooling layer, for max pooling layer, we use 2 by 2 by 2 cube. By 2 by 2 by 2 cube, what max pooling layer does is it computes, it picks up the maximum value within this box. So 2 by 2 by 2. So there's 8 values here. And then out of 8 values, you just output the maximum value. That's max pooling layer. And from here, then now it becomes, because you put a 2 by 2 by 2 to 16 by 16 by 16 box, then the size will be 8 by 8 by 8, because you put it next to each other. Then you here we put another 3 by 3 by 3. This is a convolution layer, same as size is same as before. Then it will make it to the 6 by 6 by 6. And then from here, it does another max pooling layer. And then finally, this box size is 3 by 3 by 3. And now there are, so here, the number 100 or 200 or 400 and so on, this is a number of filter we use. So for example, in the first step, we have 100 different 3 by 3 by 3 filters. And at the end, so we have 3 by 3 by 3 by 3 by 3 box multiplied by 400 of them. So that number of feature is, if you multiply 9 by actually 27 by 400, it will be 10,800. Okay, so this is the architecture. Any questions? Okay, let's start from, okay. Oh, okay, so from here, in this okay, I missed, I haven't looked at the protein, when this question is asked, okay. In protein docking, so by docking, I mean, it's a protein docking. You have two 3D structures, and then you assemble them, you combine it to make a more complex structures. What does each layer do? I think now I explained. While choosing the box, did you know the residue which would interact? Right, so here, okay, so the purpose of this DOF neural network is to predict if a model is correct or wrong. So when we applied during training, I need to know the answer of interacting residue pair. That's how we do the supervised training. But when we applied to unknown model. Why is a sigmoidal function at the end? Okay, right. So at the end, we wanted to get the probability value that ranges from 0 to 1. So that's why a sigmoidal function is appropriate. Is appropriate. May I know the reason why it's 3 by 3? Yeah. Okay. Yeah, that's a good question. It's a parameter. Well, just by looking at 20 by 20 by 20, we thought 3 by 3 by 3 is reasonable size because we want to scan it. But it's a try and error. So we don't have a. If you ask why not 4 by 4 by 4, yeah, 4 by 4 by 4 looks okay also, but it's a parameter. Why the irregular decrease in the size of the box? Okay, sure. It's not the irregular. Actually, for convolution, we use 3 by 3 by 3. For max pooling layer, we use 2 by 2 by 2. Is similar analysis done for all possible boxes? Okay. Can you specify more what do you mean by similar analysis? Okay. Meanwhile, let me go on. So is the architecture clear? I think this is a very straightforward application of CNN. General more question. Okay. So yeah, why this architecture? This architecture, I would say, is pretty regular. So so we look at the literature of in like image processing field, and then they do similar architecture. You have convolution layer and occasionally max pooling layers, and then at the end you put it in the fully connected layer. So the feature collected by convolution is put into the regular fully connected neural network and do a kind of final processing and make a prediction. This is kind of a really probably empirically explored and successful general architecture. How accurate would you say this is? Okay, yeah, I show you the results. Okay, the probability of the model. Probability of the model. The output of the output. This are you talking about this probability? This is a probability that the model is likely to be accurate, to be correct in the Capri competition standard. The filter size. What does 18, 16, 8, 3? Okay, yeah, 18. Okay, let me, yeah, 18, 16, 8. Yeah, that's a good question. Let me explain again. So the first one, 20 by 20. This 20 by 20 by 20 is a box size which I put at the interface. 18 is the results of applying 3 by 3 by 3. So, okay, maybe let me draw a picture. Can I? Okay. So, all right. So input is 20 by 20 by 20 box. And then there are, this size is 20. And then, this 20 by 20 input box is cut into 1 by 1 by 1, the cubes. So I have 20 cubes along each axis. Okay, that is what this 20 means. Now, when I put this 3 by 3 by 3 filter, so it will fit here. It's 3 by 3 by 3. And then, then when I put this box, I put the results here, right? And then now I shift this box 3 by 3 by 3 by one step. Next, but the box will be here. Then I put the results here, right? So, and then the last position of this filter is here. It's again 3 by 3 by 3. And then I put the information here. So now, if you think about it, so this is a range of the output size. Then because this is this does not have output, this does not output, the size is 18, right? 18 by 18 by 18. So that's why the 18 is the size of the output. So by applying 3 by 3 by 3, the size will be shrink 20 to 18, 18 to 16. I think it's clear, right? So then now the max pooling. What max? Okay, now I have 16 by 16 by 16. So this is 16. So then I put 2 by 2 by 2 box. And then pick up the largest value and output it. And then now in this case, I put this 2 by 2 by 2 box, not by for every 2 steps. So that gives you 8. So 16 divided by 2 gets 8. And then this is another convolution layer. So 8 goes to 6. Okay. Okay, let me answer some more questions. What was the final probability value? The probability value ranges from 0 to 1 here, and then this is a probability that the model is either correct, is correct. Probability that model is correct. What is the operation that the streamer doing, and is that user defined? Okay, so this is how the convolution does. So convolution, yeah, convolution. What convolution does is it multiplied. So the 3 by 3 by 3 box has a certain value at each position. There's 27 values in it. And let me, okay. Okay. So yeah, so let me show you this again. So here, this green box is supposed to be the, it's a data. In my case, it's a 20 by 20 by 20 box, right? this yellow one, actually yellow one is a filter, convolution filter of 3 by 3 by 3. In my case, here it's showing 3 by 3. So I think, okay, so pay attention to this, the red letters, 1, 0, 1, 0, 1, 0, 1, 0, 1. Okay, so, and then look at this, the outcome. So this, for example, 4 is computed by this yellow values, numbers, multiplied by this data value. Then it sum up to 4, right? So convolution is doing this. May I ask if I try using other activation? No, in this case, we didn't use other activation function because we wanted to get some value between 0 to 1. You could use some tangent activation function, but for example, ReLU is not the one we want to use because we want to get the value from 0 to 1. Okay, so I think, okay, now the last question is about the results. Let's see the results. Okay, so here I skipped this, but this is a criteria of considering a model is correct based on this Capri competition criteria. But just quickly, so the evaluation uses three features. So here, you see that there are two proteins interacting. So first thing to consider is at the interface, this evaluation checks if interacting amino acid residues are correct. So the fraction is called FNAT, fraction of native contacts here. Okay, FNAT. Okay, yeah, FNAT, fraction of native contacts. This is the fraction of interacting residue pairs in the correct answer, fraction of correctly predicted interacting residues. That's our FNAT. Another one is interface RMSD. Interface RMSD, this is a root mean square deviation. It's an error at the interface. LRMSD is ligand RMSD. So it is measuring if the position correctness of the accuracy of the position of the ligand relative to the another receptor protein. So these three metrics are combined, and then the model is classified into 4 classes: high accurate model, medium level, acceptable, okay level accuracy, or incorrect. So in this work in DAF, we consider acceptable higher as okay, correct, and incorrect. So it's a binary prediction, and the probability is telling that if a model is acceptable or higher. Okay, so this is about data set generation. In bioinformatics, generating the correct data set is very important. So what we did is, so there is a benchmark data set called ZDoc benchmark data set. What ZDoc benchmark data set has is, it has it has many protein complex. Okay, it has many protein complexes downloaded from PDB, and then for each of the protein complex model from PDB, it has generated models. Okay, on average, 54,000 models for each target, which that include correct one and incorrect. So, and then from this ZDoc benchmark, we used 120 targets. For each of the targets, we have about 54,000 models. The fraction of correct models within this 54,000 varies depending on the protein complex. But typically, as you can see that, okay, if this is a correct answer, and if you generate the random models, like various different orientation, the number of correct acceptable models will be much smaller than incorrect models. You can make a lot of incorrect models, but you cannot make too many acceptable models, because otherwise it's not acceptable. So to augment data, so we wanted to increase the number of correct models. For that, we did data augmentation. for data augmentation, what we did is so starting from a correct model, acceptable model, we place this protein in 6 different orientation facing in a coordinate. So we faced the proteins in 6 different orientation. If you think about the box, you can place a box like a dice. You can place a dice in 6 different ways, putting one side on the floor, on the surface, and then you can also rotate 4 rotations by 90 degrees. So from one model, we generated 24 models with different orientations. And during training, we use that. We consider we increase the number of correct models by this rotation. And so now, okay, so we increase the number of correct models in training. And we also now, and to further, but still, there are imbalance between number of correct models and incorrect models. So for each set, we sampled the same number of incorrect models as the correct models. So now it's balanced. Now, okay, so this is very bioinformatics specific data preparation. Here we wanted to remove redundancy in a data set. Out of this 120 complexes, there are some proteins which are similar to each other. In the case of well, regular machine learning, which uses image data or like a text information, like natural language processing, or the regular image processing, usually well, there's no people don't apply these steps because judging the similarity of photos, pictures are not easy. Actually, that could be another research topic. But in bioinformatics, especially in proteins, it's easy to actually, people try to remove redundancy in the data set. So because we wanted to make a model, the prediction model as general as possible. So from the 120 protein complexes, we grouped 2 complexes into one group. If at least one pair of protein from a complex has structural similarity, well, it is measured by a method called TM score, more than 0.5. So if the structure is, if at least one pair of proteins in 2 complexes are similar in structure and also by sequence, then we grouped them together. So that result in 63 groups. 120 complexes become 63 groups. So it will look like this. This is in supplemental data in a paper. So group 1 and 5 only have 1 entry. This is a PDB ID. But look, group 5 and 8 have multiple PDB entries, which means that those protein structures are very similar, or at least pair of chains are similar. And then we grouped them into one group. Now in the training, okay, so in the training, so during the training, first we split these 63 groups into four subsets, and we did a cross-validation, 4-fold cross-validation. Okay, so here, so there was 4 splits, 4 subsets of 63 groups, and then in parentheses 1 for 14 and so on. Or you see that there are lots of 35. This corresponds to this group ID. So all the proteins of the same group went into the same split. Okay. Now, in the 4-fold cross-validation, the way it works is that, okay, we take, for example, split 1, subset 1 out, and this is split 1 is considered for as a test set. Now, one is when split 1 is taken out, we used split combination of split 2, 3, 4 for training and validation. So, and then we tested on the test set. Now, okay, we use these proteins in split 1 as a test set. Now we use split number 2 as a test set. So a protein in split 2 is taken out, and then I will use split 1, 3, 4 for training and validation. And then we get results for proteins in complexes in models in split 2, and so on. So in four-fold cross-validation, we do training 4 times on different training set independently, completely independently. And when for example, when number 3 is in the is within the training set. So in that case, we used, because the number of data is not huge, in number of complexes is not that large. We only have 120. We used all of this model within the same group in training. But when we apply this model, there's a clear split between testing and training. Training data has, you can say, redundancy, but there's no redundancy between the test set and training and validation. Now, when, for example, number 3 is taken as a test set, then we apply the trained model based on 1 to 4, trained on 1 to 4, and applied the results to all of this target. But since those those PDB with the same group is kind of redundant, we averaged the results across the proteins of the same group. Okay, let's see any question. Similarity between 2 proteins can be interpreted by intermolecular force. Sure, intermolecular forces could be used, but we used the structure similarity and sequence similarity. Computing it by intermolecular forces can be a bit tricky because I'm not sure how degenerate it is. So it's it's possible that completely different complexes have similar intermolecular force values, and I don't know how the intermolecular force value corresponds to different shapes or similarity of the shapes. Would the amount of different groups affect the data accuracy? Different amount of, yeah, so in general, yes, in general, you wanted to have as many data as possible. So if you have less data, accuracy is tended to go down. Okay, so yeah, this is what I just explained. This is, we did 4-fold cross-validation. We split the data into 4 levels, and then 3 subset was used for training, one training and validation, and 1 subset was used for testing. Yeah, so all group member complexes were used for training, but in testing, the results for each targets are computed, but they are averaged. So one group only get 1 accuracy value. Yeah, augmentation, rotating the correct model into 6 different 6 by 4, 24 different poses are used only for training, but in testing we didn't consider that. Yeah, the split of during in the training data set, 80% was used training, 20% are validation. So training, okay, the difference between training and validation is that we have hyperparameters. So for each combination predetermined hyperparameter set, we train it and then apply to the validation, and then and see which hyperparameter is good on the validation set. So here, okay, so I'm showing that also hyperparameter values. This is also in supplemental data. So okay, so this is on the first column. I'm showing that the feature values, atom 20 and atom 40 means 20 and 40 indicates a box size, input box size, 20 angstrom or 40 angstrom. And then these are other features just using a statistical potential named GOP IT score or combination of them. And but you can, okay, what I wanted to show you is that the hyperparameters determined to be the best for four-fold cross-validation. So here there are two hyperparameters: learning rate, how quickly we wanted to change the parameter values in the deep learning, and L2 regularization. So this is to keep the weights in certain size. Don't want to make the weight value too large, but that's another weight for hyperparameters. The reason for each feature, the reason that we have 4 values for one feature is that we did a 4-fold cross-validation. Each of them are trained separately. So it's possible that we have very different learning, the hyperparameters for each training and for each of the 4 cross-validation. But as you can see that they're usually very similar. For Atom 20 feature, we have the learning rate of 3 cross-validations are the same. The last one is a bit different. So as you can see, actually the learning rate and L2 regularization features, hyperparameters are kind of consistent. Okay, this is showing the training set results and validation set results. X-axis with a different feature combination. Atom 20, atom 40, this is the size of the box. GOP and IT score is only using the statistical scores. A plus G, this is A. Okay, I forgot what A stands for. It should be either probably atom 20 plus GOP, G, A plus I, atom 20 plus I think, G plus I, GOP plus IT score all together. So accuracy measured for training and testing. There was some gaps, but yeah, that's how we got. So you see that small error bar. So this error bar corresponds to the 4-fold cross-validation. We have 4 values for each bar. And then this is average, and then this is, I think, the standard deviation. Okay. So yeah, this is some results. Okay, so what we did is we compared with existing methods. And okay, first, those dashed lines, all the dashed lines are our DAV methods with different feature combinations. And the solid lines are existing methods. This scope, okay, here we use scope here, but this is the original GOP scoring function. And GOP is only computed at the interface, and there's a Z rank 2, IRAD. This is also taken from this available program, but this is existing methods. What I'm showing here is x-axis is top ranks considered. So what we did is for each target have correct models and incorrect models, and we sort those models based on the probability values, and then we only look at top 5 or top 1, top 2, top 5, top 10, and top 15, and then checked if any of the correct models is included or not. If I see that correct model is included within that top ranks, we consider that, oh, we are success, right? And hit rate is counting the number of targets which have at least one correct models within that rank, top ranks. So obviously, if you see the higher value, 1.0 means that for all of the target, at least one correct model is included within that top rank. So okay, so well, yeah, we can discuss this a lot, but here I just wanted to show you that you see that dashed line on the top compared to other existing methods. The two difference between A and B is In A, I considered 120 complexes in the all in the whole data set separately. So I didn't consider the group. So in B, we have seen there are some cases that one group has many similar targets. And for each group, the proteins in the same groups are averaged. That is in B. So B is done probably more in rigorously, the rigorous evaluation, because the one group only get one accuracy value. In contrast, in A, I didn't consider the similarity groups. Maybe I should, yeah, I should skip this. This is same results, but measured in a different way. This is used, here I used enrichment factor. Enrichment factor is counting up to, when you count up to certain ranks, fraction of correct values, correct models within that rank. That is the numerator. And the denominator is total fraction of correct models within the whole data set, within the whole models, all the models of the target. So if this value is larger than 1, that means that you are concentrating the correct models within that top ranks. So by that, you see that also dashed lines are above the correct the existing methods, solid lines. So yeah, that's just using a different the metric. Okay, so we will also tried on different data set, but same story. As long as we see that dashed line higher than the solid line, we are happy. Okay, so, well, I don't have much time to go to this another approach of the same topic, but let me try to explain as much as I can. So, all right, so this is the same topic. This is a recent paper published in April, I think, this April. This is now using, this is again protein model evaluation, where I have lots of models, docking models, correct and incorrect, and we want to apply now graph neural network. Idea is the same. I have protein complex, and then so we extract the interface region, and then we represent this region as a graph, and then predict the score. So okay, I'll go detail. So in the couple of next slides, but here we first we identify the interface atoms, which is 10 angstrom within each other, between from the other proteins. So then now each dot here indicates that atom at the interface from protein in green and protein in blue. So we generated 2 graphs. In one graph, in the first graph, graph 1, I'm only connecting the graph edges within individual proteins. So those atoms are within 10 angstrom to between each other. Another graph here is now considering also those dashed line is considering the interface of atoms across 2 chains. So there are 2 graphs. 2 graphs are processed separately by graph neural network, and then finally subtracted, and then that feature is put into the fully connected neural network as usual, and then output the probability that the model is considered as correct or incorrect. Advantage over GNN over CNN. So all right. So in the earlier approach, DAV, we put a box at the interface. So actually, it is a box of a fixed size, 20 by 20 by 20 or 40 by 40 by 40. So actually, that method could not handle large protein complexes which do not fit into that box. That was a problem. But now, if you use graph, you can generate a graph for for interacting atoms. So it's more flexible and can handle proteins of different sizes, complexes of different sizes. And also GNN is rotation invariant. Rotation does not change the graph, but actually in DAV, as you see that, we put the box of 20 by 20 by 20. It can change. The performance can change by at least the feature will change if you rotate the protein complex. And also in DAV, because we put a box, some box point, like a cube, may not have information. It may be empty boxes, cubes. But the GNN, so memory, it could be memory inefficient, because in any way you need to have the big box of 20 by 20 by 20 or 40 by 40 by 40, no matter how many atoms you have in it. But a graph is more flexible, because it only considers atom at the interface. All right, so hopefully I can explain a little bit more in detail. So, okay, so we generated, so the graph, so we have graphs, and the graph is basically generated. Graph is characterized, described in an adjacency matrix. This adjacency matrix is just counting, is a matrix which tells you that which atoms are in contact closer than certain cutoff or not, 1 or 0 for this A1. For the first graph, which makes graph separately within, only consider the interaction within each protein, is described by this adjacency matrix of 1 or 0. If two atoms are connected by a chemical bond, it's 1, that pair is 1. If not, for example, this and this, it's 0. Right? As simple as that. Now, for next one, for the second network graph, which include the interacting neighboring atoms, we considered also the distance. So, all right, so this graph actually include all of the interaction in graph 1. So if two atoms are interacting, if two atoms are connected by chemical bond, it's 1. If two atoms are within 10 angstrom, then we apply this function. We compute this function. What this function does is obviously, okay, and there's a 2 parameters, U and sigma. This is a learnable parameter. But here, basically, if Dij, distance is very small, it has a high value. If the distance of two atoms getting farther away, then it will get close to 0. So it's kind of like a smooth the function between basically 1 and 0 to represent the distance between atoms across 2 chains. And otherwise it's 0. Now, okay, let's see. What if the value is changed? Okay, value means the distance. So the distance of since the purpose of this graph neural network is to judge if a static model is right or wrong. The distance will not change for a model. For a different model, they have a different distance, but for the same, because this is not dynamic simulation. So for a given model, the distance value is all fixed. Now, okay, so this is an edge. This is the information of edges. Now the features of individual atoms are like this. So for each atom, we have atom feature, atom type, CNO, S or H, and this is one hot encoding. One hot encoding means that this is a vector of 5, length of 5, and if it is C, it's 1, 0, 0, 0. 0, 0, 0, 0, 0. If it is oxygen, it will be the feature value. It will be 00100. That's called one hot encoding. Okay, so we also consider degree of atoms, connection of the atoms, also the number of connected hydrogen bonds, implicit valence electrons. But anyway, this is all the feature vector of 1 or 0. So there will be 23 features because the number of feature will be 5 plus 6 plus 5 plus 6 plus 2. That's a 23 features. But now we applied a one-layer neural network and to make it to a feature of 140 values. So one atom is represented by 140 values. So this conversion is kind of typically done. So it's making the more like a rich information has, so because the individual feature value have an information of kind of mixed information of all those features. Now, okay, so just quickly go to the algorithm. So here, now, okay, so we have a protein complex, and then we extract the interface regions, and so and then the input is the atoms at the interface within 10 angstrom to each other. So input is okay. So X is each atom, and then X is represented by this 140 features. That is the input of a graph. And then we also have this adjacency matrix for A1, just connecting edges of chemical bonds, or also consider the interface distance, A1 and A2. And these are for processing this, for the first network graph, we use A1. For processing A2, we use this the A2 matrix. And then later we subtract it, but up to some point, it's separately considered. Okay, this is the input. And now we do the graph attention. So, so now we compute the importance of connection between atom I and J. It's computed in this way. It's as you see that it's symmetrical, I and J and J and I, because we wanted to make this value symmetric between I and J. So now, okay, so what you see here is that now this X, so there was actually basically in this case, two learnable parameters. This is a matrix. X is converted, input is converted by a matrix W, weight matrix W. And then another matrix E is multiplied, and then you do it oppositely. And then that is kind of, okay, kind of like the cross product of two vectors is used, well, is representing the importance of the two atoms, atom connections. Now, all right, so the EIJ is normalized for all of the atom J interacting with I. So this is the attention coefficient. This is attention coefficient for EIJ. It's normalized, and now we also have the information of the adjacency matrix of AI or A1 or A2 defined in the previous slides. So this has the information of similarity of the features and also the interaction between that atoms. Now using that attention, the node feature is updated. So now, okay, so this is a one round of the optimization. You start from the feature, and then now you update the feature value, and then this value is considering the similarity between the features of I and J, and also the connection with other atoms. So this feature is considering the neighboring information, basically. We also add a gate mechanism. So this is updated value, and we compare. We want to combine the previous information and the new information mixed each other with weight. Okay, yeah. So there's a right. So there's a well, and the CI is defined in this way, and we apply this 4 times. And then finally, okay, so and then this optimization is done for matrix 1 and 2 separately. And then by subtracting x2 information, subtracting x1 from x2, now the information is only at the interface. Now we have the updated value for each of the node atom. And then, okay, the sum of the feature values is passed to the is considered as a feature of a graph or entire graph, so the protein complex, and then this information is passed to the fully connected neural network. This is so it has 2 things: attention and gated mechanism. Gate mechanism is just basically just combining previous information and current information together. Okay, I think I'm running out of time, so I just go very quickly. So we have same as before. We have data set. In this case, I'm showing the different data set, but we cut it into fourfold cross validation. We also did the redundancy removal. Well, dash line is our method is going higher than the existing ones, which made us happy. Okay, yeah, just let me just go through 1 minute for Cryo-EM. I could not, well, I could not go through didn't have time to go through it. Our lab is also working on Cryo-EM data. Cryo-EM will give you will give you the 2-dimensional information, and then from that, by stacking 2D information, you get the 3D information. And from 3D information, we want to guess model the 3D structures. So the input is a low resolution. Okay, this is a statistics of EMDB. EMDB is the database of electron microscope. So idea is that, okay, so we have now the input is the Cryo-EM data. Same as before, we've scanned this EM map. It's a 3D volume data and density values. So it's a grid data actually. At each grid point, you have a density value. If the density value is high, there's atom, probably some atom should be there. If the density is low, probably nothing is there, or atoms are far from that point. So by scanning it here, okay, well, I really skip it, but this is using ResNet. This is a little bit advanced, more recent architecture of the deep learning network. And by doing that, we want to predict what in that each what exists, what in that each box. So here, for example, this is detecting. Okay, on the left side, this is the experimentally determined structures. But by scanning the Cryo-EM map, we can identify alpha helix in red. Okay, this is DNA in cyan, the beta strand in yellow and green loops. So as you can, if you compare them, they are really accurate. So by scanning this low resolution image by a box, we can predict, we can identify, classify that box into alpha helix, beta strand, or other part of protein, or DNA or RNA. It's another kind of a straightforward application, logically at least, to 3D image data. Okay, I skip all this. And we can also use that detected alpha helix information or beta strand information or our atom type information in structure modeling from Cryo-EM map. Yeah, we can also make a super resolution EM map, improve EM map. So this is, okay, this is experimental data, low resolution data. By using deep learning, we can sharpen the image using GAN architecture. A GAN architecture is used for example, making a fake well, artificial image of human faces. So GAN can learn how the real sharp image should look like, and then starting from the low resolution raw data, we can sharpen it. Yeah, I need to skip all this. Yeah, if you're if you're interested, go please visit our lab website. Yeah, you have publication, and there are many software. Especially currently, we are working on Cryo-EM modeling. And this is our lab member from sometime. Well, the participants are not full member here, but next speaker is Charles from our lab. This is another picture. Okay, Charles is here. And yeah, thank you for your attention. Any last questions?
Yeah, thank you so much for so many information.
Oh, okay.
Your up-to-date research. So it's, yeah, thank you so much. A lot of links to you, and people can find your publications.
That's one question about moonlighting proteins. Yeah, moonlighting proteins are interesting and multifunctional proteins. We have worked on moonlighting proteins. We predicted moonlighting proteins from features. If you're interested in, please look at our paper, our lab publication list. Can protein docking and model selection be used for predicting multifunctional moonlighting proteins? So, okay, do you think, okay, moonlighting protein might have multiple different poses, docking poses? That's an interesting idea. That could be interesting. But the moonlighting proteins do not necessarily, okay, have multifunctions by different docking poses of the same protein pairs. Maybe they interact with some other proteins to make to realize multiple functions. But so if the interaction partner changes, yeah, we need to know which one is what is what interacting partner is. Okay, all right, okay, let's see. CNN or ML in general? Yeah, okay. So CNN, yeah, that might be possible because all right. So sure. Okay, yeah. So to use CNN or machine learning to predict if a sequence, artificially generated sequence, can structurally possible or not possible. I think it's in generally possible, but the difficulty in machine learning in general is to get a good data set. So if you have some data set of, if you have a known set of structurally possible proteins and actually impossible proteins, then using that, you can train deep learning. So, okay, in PDB, there are so many proteins, all the structures in PDB are structurally possible proteins. So if you have a list of sequences of structurally impossible sequences, then we can do the training. Okay, yeah, thank you for the attention. Okay. Apply this technique. Genetic diseases. Okay. Yeah, sure. Yeah, right. Sure. Genetic diseases. Sure. Yeah, we need to formulate it in some way. Protein geometry features. Yeah, sure. GAN. Yes, GAN. Yeah, GAN can be applied to in protein geometry features. Yeah, probably GAN may be able to apply to even probably refine protein structures. As long as you have data, machine learning can do a lot.